Yeah, recording is started, sir.
Okay, great.
Oh, thank you.
Welcome to ASTHI webinar series. Our speaker today is a young researcher and her name is Vianney Chopra. She is from the Institute of Nanoscience and Technology, Mohali. Vianney earned her bachelor's degree in pharmacy from Punjab Technical University, and her master's degree is in pharmacy with a specialization in pharmaceutics from JSS University in Mysore. In July 2017, Vianney began working as a research fellow in Professor Deepa Ghosh's lab at Institute of Nanoscience and Technology. Vianney is engaged in the design and development of novel materials and scaffolds for use in tissue engineering. She is now maybe in the final phase of her PhD study and would share some intriguing research findings with us. With these few words, I welcome Vianney to give her presentation.
Thank you so much, sir, for the introduction. Good afternoon, everyone. I am working on ceramic and carbon-based materials in the field of bone regeneration. So let me begin. I'll start with a brief introduction about what bone defects have been causing to the general population at large. The global burden of diseases has estimated that that the number of fractures in the world has drastically increased to around 33.4% over the past 2 decades, and a lot of revenue is being spent on the bone related products, for example, pins, etc. As well as the causes of bone defects goes, it can be due to a variety of reasons, for example, age, trauma, and pathophysiology, out of which different disease states like tumor, osteomyelitis, and osteoporosis have been known to be the primary causes. When we look at the structure of bone, we can broadly define it as the long bone and as a flat bone. So depending on the structure, we have different cells which are usually maintaining the formation and the modeling and remodeling of the bones, and these the osteocytes, osteoblasts, and the osteogenic progenitor cells and the osteoclasts are the main cells that are responsible for the bone reformation. So whenever there's a bone defect or bone injury, there's always a development of a hematoma, which in turn leads to the cascade of various pathways which leads to the healing of a bone naturally, which involves the activation of the progenitor cells, the active resorption of the existing bone, then the formation, the reversal, and as well as the formation. This is a general mechanism for the healing of a healthy bone. But what happens when the bone loses its ability to heal on its own? This happens in the cases like we discussed earlier, like in the case of a critical size defect, in which the size of the defect is more than 50% of the injured bone, and the bone on its own cannot heal. As well as in the cases of tumors where a large portion has to be resected, and also in the cases of osteoporosis because it leads to low mineralized bone. So the current treatment regimen that is generally being applied since the past 100 years is basically dependent upon the gold standard, which is using the autograft. That is, you take the bone from the donor himself, basically the pelvic bone, and then you implant it. Then we have then the other treatment regimens include the allograft as well as the xenograft. And recently the bone graft substitutes like demineralized bone has also been used, but all of these have certain limitations. As in the case of autograft, we have low availability and there's a donor site morbidity, as well as in the other cases we have chances of immune rejection. The processing of these grafts leads to the reduction in the osteogenic factors, as well as their chances of disease transmission taking place. So this has led to the development of bone tissue regeneration field and the approaches used in it. So coming to bone tissue engineering, the basics of bone tissue regeneration follow the triad, which is the use of cells, use of bioactive molecules, as well as use of scaffolds for the regeneration of bone. Cells vary from mesenchymal stems to a variety of MSCs, which can be obtained from various sources. But the disadvantage of cells are that they are highly immunogenic. The culturing techniques are also very expensive, as well as they can be invasive. When it comes to bioactive molecules, they have largely been used. For example, we have a lot of recombinant BMPs that have been used, but the disadvantage that has been found is that they can cause local toxicity, they can cause osteoporosis and different problems. And the scaffolds that we use are basically based on various hydrogel based or composites or ceramic based materials, and people have used this in combination with cells or with bioactive molecule or using all of them together. Like when I was talking about the disadvantage of bioactive molecules, like the molecules which are clinically available and have passed the class 3 trials, a lot of molecules are being withdrawn due to these disadvantages. So this leads to the development of newer techniques which can bypass our bioactive molecules. A brief history of time of bone regeneration shows that how we have moved from bioresorbable metals and calcium phosphate materials to the 3D printing technologies that are currently being applied. So with this goal, our basic objective for any scaffold that we want to use for bone regeneration was based on these 5 factors. Our first factor was that it should be osteoconductive. It should be able to support the growth of the new bone, the scaffold. It should be inductive, osteoinductive, so that it can recruit your osteoprogenitor cells to that area. It should help or aid in the proper integration with the surrounding bone. It should also be angiogenic so that it can provide the proper vasculature for the nutrients as well as for the cells. And it should also have an inherent antimicrobial property, because in case of implants, there are chances of infection. So keeping these 5 goals in mind, we have used a technique based on the different requirements. Which are based on in bone tissue engineering. So the biological requirements are that it should be biocompatible, it should be non-immunogenic. So based on this, you can go for the different structural features that are required, which are bio-inspired. It should have a higher porosity, it should be customizable. So the composition that generally are being used are based on ceramic or polymeric composites as well as carbon-based materials. With different manufacturing techniques have also been used, like freeze drying, gas forming, and electrospinning, as well as 3D printing. So with these 4 requirements, the basic aim has to go for an acellular and a growth factor free approach, so that the scaffold that we have can aid in the cellular attachment as well as the different properties and the different goals that we have. So we have chosen ceramic based materials in which we have gone for hydroxyapatite, as hydroxyapatite is present in at the nano level structure of the bone. It is present in between the collagen nanofibrils, and these hydroxyapatite particles are known to provide to give the strength to the collagen fibrils when it forms its osteins and everything. And at the nano level scale, nano hydroxyapatite has been known to increase the ossification as well as cellular addition and proliferation. And a lot of new reports have shown that when we add metal ions like zinc, strontium, cobalt, etc. to it, it has known to improve the osteogenic properties of this nanohydroxyapatite. So based on the literature, we chose nanohydroxyapatite. And we have gone for carbon-based scaffolds because in the past 2 decades, a lot of work has been published on graphene and RGO, reduced graphene oxide based materials for bone tissue regeneration, because they have a large surface area. They can bind a variety of physiological molecules and proteins to it. They have exceptional electrical as well as mechanical properties, and they have also been known to be osteoinductive. A lot of literature has reported the differentiation of MSCs into bone as well as various other lineages. So with this goal, literature, we went for the carbon-based scaffolds. So our basic goal was to develop a scaffold which would be able to provide osteogenesis by osteoinduction, osteoconduction, angiogenesis, as well as provide a good osteointegration with the required biophysical as well as the chemical cues for a good bone regeneration. And here we have gone for an acellular growth factor free approach using functionalized carbon as well as ceramic biocomposites. So our first approach was the development of a silver nanoparticle based hydroxyapatite and reduced graphene oxide gelatin cryogels. We have chosen silver nanoparticles for their broad spectrum antimicrobial activity because they are known to produce oxidative stress in antimicrobials, and they are FDA approved also in a lot of cases. We went for hydroxyapatite due to its osteoconductive property, and graphene oxide was chosen due to its osteoinductive property. We have selected cryogels as a scaffold for this material because cryogels are formed by freezing our liquid, our gels, the polymer state at sub-zero temperatures, and then we thaw it and then we freeze it again. So this leads to the formation of ice crystals, and after the thawing, it leads to the formation of large voids. So we have a large macroporous network being formed, which will aid in the migration of cells as well as in the formation of vascularization. So for this, we have chosen gelatin as a polymer. So when we come to the synthesis, our first step was the synthesis of blank hydroxyapatite as well as silver nanoparticle doped hydroxyapatite. This was done using the required calcium and the phosphoric acid sources, and we added silver nitrate solution to it, and after stirring we transferred it to a hydrothermal, and this reaction was kept for at 180 degrees centigrade for 18 hours at an alkaline pH, and we observed and we characterized it using XRD studies, which showed the formation of silver nanoparticles on hydroxyapatite. This was further characterized using TEM analysis, in which we can see that 8 to 9 nanometer size of silver nanoparticles are formed on nanohydroxyapatite rods. This was extensively characterized using FTI as well as XPA studies to find out the state of silver in these nanohydroxyapatite rods. Our second step was the formation of the cryogels, in which we have chosen gelatin as a polymer. We have mixed gelatin as well as GO together and heated it for 70 degrees for 24 hours, after which we have subjected these to further freezing and thawing cycles using a lyophilizer for at minus 20 degrees centigrade. So after optimizing the various different cycles and different concentrations of GO and gelatin, we were able to form a highly cross-linked cryogel after 5 freeze-thaw cycles, which was optimized. And this cross-linking was taking place due to the chemical cross-linking, and secondly due to the physical cross-linking taking place between the OH groups and the H groups, NH2 groups of the gelatin as well as the RGO present. And an interesting result that we saw was that when we were doing this, when this cryogel formation was taking place, we saw that there was a reduction of GO taking place to RGO, which aided in the further enhancement of the cross-linking. So this approach shows that we are using a cross-linker free approach to develop cryogels, because usually the cryogels that are developed, like the gelatin cryogel, which is available in the literature, shows that it uses glutaraldehyde or other cross-linkers for the cross-linking, but these cross-linkers can be toxic when applied at the biological sites. So we have gone for a cross-linker free approach. So we characterize these cryogels after incorporating various concentrations of silver nanoparticles, hydroxyapatite in these. So 1, 2, 3, and 4% were chosen, and we formed the morphology. We saw that in the case of low loading, we had higher porous network, and this was collapsing in the cases of higher silver hydroxyapatite concentrations. We characterized it for porosity, swelling index, and mechanical strength, and we saw that the 2% loading of gel RGO showed good properties of mechanical strength as well as the porosity. We carried out the degradation in a collagenase solution because for a scaffold to be present in the body, it has to be there for a longer time. So we saw that the degradation was slow in the case of all these 4 concentrations, and it was for over a period of 3 to 4 weeks. Then we carried out the silver release that was taking place from the cryogels, and we had a continuous release at for a longer period of time. And we finally optimized, and we said that okay, we had a good porosity, these cryogels had slower degradation in comparison to the gelatin cryogel, and we had longer silver releases as well as a good mechanical strength was also obtained. Our second study was the cytocompatibility, because higher doses of silver can be toxic. We had to see what concentration to use further. So using the basic cytocompatibility studies on L929 cells, we saw that 2% was not showing much decrease in viability after 48 hours, whereas 3 and 4 was slightly decreasing the highly decreasing the viability of cells. So we chose the 2% for the further studies. We carried out the hemocompatibility studies of these solutions, and we saw that our gelatin and the other cryogens were not being toxic to the RBCs. Our next study was to carry out the antimicrobial study. So we incubated our cryogels with SRES over a period of various time durations, and due to the continuous release of silver, we saw that there was a significant antimicrobial property also obtained for these cryogels. So we saw good hemocompatibility and a good antimicrobial activity. Our next study was to carry out the osteogenic potential of these cryogels, and we used MG63 cells initially, and we saw that the cells were getting attached on the surfaces, and as well as there was an increase in the there was an increase in the proliferation as the cells are proliferating. As you can see that the porous networks are the number of density of the cells has increased significantly. Next study was to carry out the alizarin staining to see how the mineralization nodules were being formed on these cryogels, and when after extracting the alizarin dye, we saw that there was a higher concentration for the silver hydroxyapatite cryogels. The next study was to see how the genes are being upregulated by the hydroxyapatite containing cryogels and the RGO cryogels, and we saw that there was a significant upregulation of COL1, the initial marker, as well as OPN, which is a late marker taking place. So we saw enhanced cellular proliferation, addition, and osteogenic activity. The only disadvantage that limitation I found out of the study was that the high release of silver was not helping my endothelial cells to attach, and it showed a limited angiogenic response. We did publish this paper, and we were able to formulate a good cryogel which showed supported osteogenic properties using silver nanoparticles and hydroxyapatite. So our next study was, because we had begun with those five main parameters, we did not get angiogenesis in the study. So we decided to change our ion, and we went for a zinc doped hydroxyapatite approach and an RGO nanocomposite approach. So we have chosen zinc in this because zinc has been reported to be angiogenic as well as antimicrobial, osteoinductive as well as osteoconductive. So we synthesized over here zinc doped hydroxyapatite using the hydrothermal synthesis, and we characterized it using XRD, and we saw that in a similar way, when we added graphene oxide to this hydrothermal synthesis, we saw that there was a reduction taking place, that GO was getting converted to RGO, which was characterized by Raman analysis as well as using XPS analysis. The deconvolution spectra of carbon 1 and Zn show that there was a presence of zinc ions, as well as there was the reduction of the CO groups was getting reduced for the final composite. The SEM analysis showed the formation of hydroxyapatite nanorods on the surface of RGO, which was further characterized using HRTM as well as SAED spectra. We carried out the water contact angle properties of this material because GO is slightly RGO is known to be hydrophobic, but we saw that plain for this comparison, we even synthesized plain hydroxyapatite on RGO surfaces without zinc to have a comparison how zinc was playing the role. So we saw that plain hydroxyapatite with zinc and the GHAP, they both had the required water wettability property that is required for the scaffold for the cellular attachment. We saw a continuous release of zinc taking place for over a period of 60 days from the zinc hydroxyapatite composite, and also we carried out the protein adsorption studies, and we saw that the presence of GO, there was not a significant change, but GO was the reason that there was that addition of hydroxyapatite to GO aided in better protein adsorption. So we saw optimized zinc doping, and we saw a continuous release of zinc taking place. Our next study was to carry out the antimicrobial property of it. So we saw that when we coated our substrates with our material, we saw a good anti-biofilm property. We saw a larger number of dead cells on the surface of zinc containing composites, and which was optimized with the CFU inhibition of plating technique. The hemocompatibility studies were also carried out, and we saw that that the composites were not showing any toxicity, and that they were biocompatible as well as hemocompatible. Our next study was to carry out the osteogenic differentiation of MSCs. Here we have isolated mesenchymal stem cells from umbilical cord of human samples, and we have propagated them in the third and the fourth passage, and we saw we went for the MTT studies, cell proliferation as well as attachment studies, and we saw that there was a there was slightly improved proliferation rate in the case of zinc hydroxyapatite containing RGO composites. We saw the cellular attachment taking place on the surface of these substrates. Allicin and red staining was carried out to check the formation of calcium nodules taking place on the surface of the cells, and there was an increased calcium mineralization taking place for these zinc doped hydroxyapatite nanocomposites. We carried out the qPCR studies to see how the genes were being upregulated, and we saw that there was an induction of BMP2 in the earlier days, and OPN and OCN was getting increased at a later stage. So our composite showed good attachment proliferation as well as osteogenic differentiation. Our next study was to perform the angiogenesis studies, and over here we have isolated human endothelial vein cells from the umbilical cord, and we went for the cellular viability studies as well as the attachment studies, and we saw that zinc was aiding in cellular zinc release was also aiding in the proliferation as well as in the attachment. We saw increased levels of FGF as well as PTGF, which are responsible for endocellular recruitment as well as attachment of the cells. There was better vascularization taking place for these cells on a matrigel assay. So we saw that the zinc release from these nanocomposites showed an upregulation of the angiogenic markers as well as it was aiding in the angiogenic profile, endogenic nature of the cells. Our next study was to check out how they were behaving in the in vivo system, and over here we've created a 0.8 mm defect in the rat femur, and we have implanted our material, and we saw that over a period of 2 weeks there was increase in the bone volume, as well as when we injected calcium we saw that there was a higher recruitment of cells taking place. The matrigel plug assay was carried out to check the angiogenic profile of it, and we saw that in the case of zinc there was an increased level of hemoglobin concentration. So we saw that the zinc doped hydroxyapatite RGO composite was aiding in the callus formation as well as the cellular recruitment and as well as the vascularization. So we were able to achieve the properties that we had initially gone with. We had osteogenesis, we had angiogenesis, antimicrobial, and an antimicrobial property. So we achieved it, and we have published this study also. But the limitation I found out was that when we put it in a delivery, we needed a delivery system for this composite. We had added it in a alginate gel, and we found out that it had a tendency to be washed out easily. So in order to improve our material, we decided to go for a delivery based approach for these systems, and we went for a gelatin nanofibers to incorporate our zinc doped hydroxyapatite and RGO composites in it. So our next study was based on this previous study, where we had optimized that zinc hydroxyapatite had a good biological activity. So what we wanted to check in this study was that if we have zinc doped hydroxyapatite as such, and if we incorporate the zinc doped hydroxyapatite on the surface of RGO systems to a gelatin nanofiber system, how it would behave, and how it would behave even in the absence of any external growth factors if we added to the in vitro stimulation system. So for this we went for electrospinning technique, and we chose gelatin as the polymer for nanofibers. Our first step was for the synthesis of zinc doped hydroxyapatite as well as the formation of synthesis of zinc doped hydroxyapatite on RGO surface, and then the further incorporation of this in the gelatin nanofiber systems. We optimized our nanocomposites using TEM, XRD, and the XPS and other studies, and then we incorporated it in the nanofiber system, and these were the nanofibers that were being formed. So our nanofibers were then further characterized for their compatibility studies, which are important for cellular attachment as well as biomolecular attachment, and there was a slight increase taking place. The zinc doped hydroxyapatite with gelatin nanofibers were hydrophilic. The addition of RGO to this slightly made these fibers hydrophobic, but it was still in the acceptable range, which is required for cellular attachment. Our next study was done in simulated body fluid, in which we wanted to check how our nanofibers would behave. And after 28 days, we saw that there was good biomineralization taking place on the surface of these nanofibers, that they aided that more calcium calcium nucleation was taking place, and this aided in the calcium mineralization. So we went for the mechanical properties next. We saw that there was an increase in the elastic modulus as well as the tensile strength of the RGO containing composites, which was, and even in the case of hydroxyapatite composite, it was improved in comparison to the plain gelatin nanofibers. We saw continuous zinc release taking place from these nanosystems, and because the next study we did was the degradation studies, and we saw that because gelatin nanofibers showed a higher rate of degradation. They were not they were almost degraded after 30 to 40 days. But in the case of the incorporation of these nano composites to the gelatin nanofiber system, we saw a reduced degradation rate. We saw that around 4 to 5, they were still viable after 4 to 5 months when we kept it. So we optimized the wettability, and we saw that there was an increase in the mechanical strength, and degradation was slowed, and biomineralization was improved. The next study was the antimicrobial property and the biocompatibility. We incubated our materials with bacteria, and we saw that there was a continuous reduction in the colony forming ability because of frost generation taking place due to the release of zinc ions. We saw that on the surface of the nanofibers, the bacterial cells were not able to survive, and it showed an anti-biofilm property also. Our hemocompatibility studies showed that all the nanofibers were hemocompatible, and as well as they were cytocompatible. This study has not been shown here, but we have done it. Our second study was to check the osteogenesis in a growth factor-free media, because this was our main approach. So our first study was to check how our MSCs were behaving in the presence of these nanofibers. We seeded our UCMSCs on the surface of these nanofibers, and using actin and HES staining, we saw that there was a good attachment taking place in the case of the zinc hydroxyapatite with RGO nanofibers, with gelatin nanofibers. As you can see in the SEM studies also, we can see the protrusions and the filament attachment taking place over here. Our next study was carried out to check how there was the qPCR studies, that we checked both in the basal media, which did not have any osteogenic supplement to it, as well as in the osteogenic media, to have a comparison of how the release of zinc and these nanofibers, the biophysical and the biochemical cues were behaving, like the cells were behaving in the presence of these cues. And we saw that even if you take in the basal media also, there was an upregulation of RUNX taking place, as well as there was an upregulation of OCN taking place in the later at the later time points. And there was no significant difference between the basal and the osteogenic media. Although the osteogenic media had a higher value, but the significant difference was very low in the case of the zinc HP and RGO gelatin nanofiber systems, which was further confirmed using in the immunofluorescence studies. Like for in the basal media, when you look at the gelatin nanofiber systems, for call 1, you could see that there was less fluorescence, which in the osteogenic media was significantly higher. But in the case of zinc HP with gelatin and zinc HP RGO, we saw that the immunofluorescence was comparable for in both the basal media and the osteogenic media. Similar results were obtained for the OCN studies, OCN immunofluorescence, which showed that even in the absence of the osteogenic media, the basal media and the zinc ions and the presence of these nanofibers aided in the MSC differentiation. So we saw a significant osteogenic differentiation of MSCs even in the basal media without any growth factors. Our next study was for the angiogenic potential. Over here, we saw the migration of Hewitt cells taking place in the presence of the extracts of these nanofibers, and we could see that the scratch was closing due to the release of zinc. And as we can see over here, there was no difference between zinc HP and RGO, which showed that only zinc was playing a role in the migration of the cells. The next study was the gene regulation, and we saw that there was an increased upregulation of INOS and FGF, which is responsible for the angiogenic potential. We further characterized this using CD31, which is a PCAM molecule, which helps in the addition of endothelial cells. And we saw that in the case on the surface of the gelatin nanofibers, which had RGO in it, there was an increased CD31 fluorescence, which can be correlated with the increased FGF levels. That showed that there was an increased addition of the cells on these nanofiber systems. So we saw improved antigenic response on these nanofibers. The next step was the in vivo biocompatibility and the vascularization. We used a subcutaneous pouch model in which we implanted our materials. This is a defect area, and we could see that there was a higher there was a higher migration of cells taking place over here, and as well as a new we could see that the CD31 expression for in the case of our final form nanofibers was also increased due to the release of zinc, as we saw in the in vitro studies also. So we saw that there was an increased cellular activity and good vascularization potential, which would aid in the early stages of healing. Uh, we went for the 5 different properties that we had initially aimed for, and we did achieve all those properties, and we were able to publish this study also. The limitation of this study was that it can be although it can be used for the cranial maxillofacial surgeries which do not require that much mechanical load on it, but the limitation was that it could not fit in irregular defects in cases of larger trauma. So we are working on improving our material, because they will not this is the limitation with our specific with fixed geometry scaffold, that they will not be able to fill the defect sites properly. Then they will be highly invasive, when we have to apply a cryogel to any defect site, it is highly invasive. So we are looking forward in the area of injectable bone cements which can fill irregular bone defects. So our ongoing work is based on calcium sulfate based bone cements, and we are trying to improve its limitations, which are faster degradation, and it does not have any inductive properties, mechanical strength, and the fixation issues which are greatly present. So we are trying to increase all the change these properties and incorporate all these different properties in our calcium sulfate based cements. We are using a gold nanoparticle based approach, which is currently undergoing. So my conclusion that I came from all these studies was that we can incorporate various metallic ions to hydroxyapatite and RGO composites, and they show they show the required biophysical and biochemical properties which are required for osteogenesis as well as angiogenesis. We were able to improve the mechanical properties of the existing scaffolds, and we saw good antigenic potentials of the zinc iron doped hydroxyapatite in both in vitro and in vivo models. And we look forward to taking our materials further, like we can use it to coat our metal alloys in case of critical size defects. We can use these to print customized 3D printed scaffolds, as well as we are using the cryogels that we have developed for other tissue regeneration applications. So this is what I've been working on, and I'm looking forward to work on in the future too. I would like to acknowledge my research supervisor, Professor Deepa Ghosh, as well as Director INST and INST for providing me the funding, as well as collaborators, especially the ST group and Professor Naivita Chattopadhyay and as Dr. Rajiv Guha sir for the help in the animal studies, and Dr. Pradeep Saha from PGIMR for providing us with the human umbilical cord samples. Thank you.
Okay. Nice and lucid presentation. I think you may need to turn off you have to mute. Yani, if you can mute. Yeah. Okay. Okay. Something has gone wrong. Am I audible?
Yes, sir.
I'm audible. Okay. Okay. So I'm sure that people will be having some questions. This is a very interesting set of data. Particularly the challenging aspect I find is to have any implant which is acellular as well as growth factor free. In this regard, if you go to your slide number 35, there I have a clarification. Now, here, when you say that you have done these, for example, take the first left-hand panel, how did you induce, or maybe when you are showing immunofluorescence of collagen and osteocalcin in the right-hand side. So their growth factor free, by that do you mean that it has no differentiation, means no, it doesn't have any ascorbate and beta glycerophosphate? Is that your growth factor deficient means? That it is devoid of those two compounds?
Yes, sir.
Okay. Now, why would you call them really, do they come really under, strictly speaking, growth factor? Beta
No.
glycerophosphate, ascorbate. I mean, growth factor one, you would assume that, okay, you are adding BMP2, for
Yes.
example.
Yes, sir. It was
So,
a exogenous media free approach, like TG.
right, you did not induce differentiation by giving actually too much of phosphate, means beta glycerophosphate.
No.
You have avoided it. So, but that makes it quite interesting, is that you are using a regular growth medium, and your presence of this material is able to induce differentiation, which suggests that it is a pretty strong or potent differentiation inducer, right?
Yes, sir.
It is a potent differentiation inducer. Now, another clarification, when you have done extensive characterization of your materials.
Yes, sir.
Now, have I missed the characterization of the mechanical properties? For example, shear strength or tensile strength, that kind of fatigue strength of the material?
So we did the tensile strength of the material for
You
these
did.
samples. Yes, sir.
Okay, this has been, that has been done. Here. Somehow that is what I was forgetting. Okay, you have done. So how did you do this thing? Elastic modulus and tensile strength?
Sir, we have a BIS system over here. So we attached our nanofibers to the two ends, and then we have a 200 Newton force over here. So we did it using that system.
Okay. Please.
So we were checking how the elongation was taking place and at what point it was breaking.
Okay. Now, in terms of, I don't know, what are the standard regulatory guidelines for orthopedic implant with modified metallic surfaces for bone or bone cement? Is any regulatory guideline issue is being unaddressed
For sir?
for if you want to develop for clinical translation?
Yes, sir.
For clinical translation, I don't know, I'm not knowledgeable about the current regulatory guideline regarding implants for with modified metallic surface opposing bone or bone cement. So when you are doing these development studies, do you have any deficiency in any parameters that needs to be done so that we are aware of it?
Yes, sir. So the deficiency that I have in my study was that we need to still check how in the long run, because reduced graphene oxide and graphene oxide, although they are very good materials, but they still face some regulatory issues. So what we have to check is in how in a long-term study, like for over a period of 1 to 2 years, how our materials are behaving in vivo system. We still need to optimize how the scaffold is going to get degraded in vivo system. In vitro studies, we have characterized extensively, but how it behaves in a higher animal model and what is the different profile of bone healing and the integration taking place, that is what has to be checked for its further translation. We are still in the very initial stages for these materials.
Okay, so I'm done with clarification. Let others make any comment or question, suggestion, please.
Coming to the question you had asked regarding the use of these materials for clinical application, there
Yeah,
are
please.
certain, so there are reports which says that graphene oxide RGO is being used for in some systems they are showing that these could be toxic, and in some systems they are showing that in long term effects there is no toxicity. So the jury is out as such. We really do not know what is the long term effect of using this material. So as Vianney was mentioning, we really need to do a long term study and also to see whether it is retained, or during remodeling does it get thrown out. So these are some of the questions that are yet to be addressed.
Another thing I don't know about bone implant, but this much I know, that in case of any osteogenic therapy, one have to do safety in terms of osteogenic sarcoma. So since here your aim is to induce bone regeneration, which is the other side, or which is in other words one would say osteogenic stimulation, wouldn't you need to in long term, wouldn't you specifically need to see that whether or not it is causing osteogenic sarcoma? Is the regulatory body clear about it?
No, sir, it isn't.
No, because it will stay there. It will stay there for till it is not completely remodeled and out of the tissue. So it will stay there for a long duration. So the issue of, maybe because it's a localized versus when osteogenic therapies are given, it is given systemically. That may be the reason why this concern is not there. I think that may be the reason. Right.
Yes, that is true. What you are
That may
saying
be
is
the reason.
absolutely true.
Right. That may be the reason. Right. Yeah. And finally, how do you address another major orthopedic challenge of giving some implant at the site of the spinal injury? Spinal injury. Did you hear what I say?
Yes, sir.
Yeah. Okay. That is a major challenge, that if you have a major spinal injury, what kind of implant to be used?
Yes, sir. So we were looking forward to injectable bone cements, but still for spinal injuries, for it to take the exact shape and everything is still a challenge. So either we can go injectable, it will still be a disadvantage. So we can go for customized 3D printable scaffolds for it. So that is the
Injectable is still there.
It is still there,
Injectable is
but
still
then
there. Yeah, yeah, it is there for a long time now. Yeah.
it is there for a long time. So we are trying to, our latest study is based on that. We are trying to incorporate different properties to the injectable cements so that they can be retained for a longer time and as well as they can have improved properties.
Okay.
We're trying to provide a mechanical stimulation to these cements.
Mm. Any other
That
question?
has,
Yeah, please. Yeah.
sorry, that work she has not presented here because I think she thought it would might take too long. Where she has developed an injectable cement, which also has both osteoinductive, conductive properties. Normally,
Mm.
such and also the mechanical strength has been improved. So that particular study would probably give us an idea how to take it forward for both 3D printing bone structures as well as even looking at it in terms of injecting the material into spine and see if how it can help in the regeneration process.
Yeah, sure. Any other question? If there is no other question, then let me again thank Vianney and congratulate her for this such a highly interesting and exciting work. And the congratulation is also for her PhD mentor, Dr. Deepa Ghosh. And we hope to work together and learn more and more exciting things. And I will make sure that Shivendu, for some reason, was unable to join, but I will share the YouTube video link with him, and I'm sure that you two have good collaborative potential. So wish you all the best and prosper in your career, Vianney.
Thank you so much.
And thank you very much, Dr. Ghosh. See you soon.
Thank you. Thank you. Yeah.
Sure. Bye.
Have a nice weekend. Bye.
You too.